Was it moldy bread that helped discover penicillin? Knowledge shall increase. Series 1. No, moldy bread did not discover penicillin, but rather it was a fungus similar to the one found on bread that led to the discovery of this antibiotic. Scottish bacteriologist Alexander Fleming discovered penicillin in 1928. Fleming observed that a mold grown in a culture of bacteria killed some of the bacteria. The mold was a strain of the fungus Penicillium, which can be found in soil, grain, rotting fruit, and elsewhere. With the use of penicillin in the 1940s, the era of antibiotics began, and this milestone has been recognized as one of the greatest advances in therapeutic medicine. The discovery of penicillin and the initial recognition of its therapeutic potential occurred in the United Kingdom. However, due to World War II, the United States played a crucial role in developing large-scale production of the drug. This process ended supply constraints and allowed penicillin to become widely available and begin saving lives. Alexander Fleming and the discovery of penicillin. Penicillin ushered in the era of antibiotics. Before its discovery, there was no effective treatment for infections such as pneumonia, gonorrhea, or rheumatic fever. Hospitals were filled with people with blood infections, contracted from a cut or scrape, and doctors could do little for them except wait and hope. Antibiotics are a compound that bacteria and fungi produce naturally to kill or inhibit rival microbial species. This phenomenon has long been known and may explain why the ancient Egyptians applied a poultice of moldy bread to infected wounds. But it was not until 1928 that Alexander Fleming, a professor of bacteriology at St. Mary's Hospital in London, discovered penicillin, the first true antibiotic. Before he went on vacation, Fleming was working with colonies of Staphylococcus, a bacteria that causes boils, sore throats, and abscesses, and decided to pile up the petri dishes with the cultures in a corner of the lab. Was it a coincidence, or did the apparent disarray of his lab help him discover the vital weapon against infections? Upon returning from vacation on September 3, 1928, Fleming discovered that one of the cultures had been contaminated with a fungus, or mold. The area around the mold, later identified as a rare strain of Penicillium notatum, was clear, as if the mold had secreted something that inhibited bacterial growth, and around it was an empty area where the Staphylococcus colony had been destroyed. At this point, he began sorting through petri dishes containing Staphylococcus colonies. Fleming found that his mold juice was capable of killing a wide range of harmful bacteria, including Streptococci, Meningococci, and the diphtheria bacillus. He then assigned his assistants, Stuart Craddock and Frederick Ridley, the complicated task of isolating pure penicillin from the mold juice. It turned out to be a very unstable compound, and the researchers were only able to prepare impure solutions, but they were useful for further work. Fleming published his findings in the British Journal of Experimental Pathology in June 1929, making only a passing reference to the potential therapeutic benefits of penicillin. The program of experiments and clinical trials required the team to process up to 500 liters per week of mold filtration. They began growing it in an odd variety of vessels, including bathtubs, chamber pots, milk churns, and food tins. A fermentation vessel was then designed to facilitate removal and, to save space, to renew the broth below the surface of the mold. They hired a team of penicillin girls, paid £2 a week, to inoculate and tend the fermentation process. The Oxford laboratory became a penicillin factory. Albert Alexander became the first recipient of Oxford penicillin. He had scratched the side of his mouth while pruning roses and developed a life-threatening infection with huge abscesses in his eyes, face, and lungs. He was injected with a dose of penicillin, and within a few days he made a remarkable recovery. Unfortunately, supplies of the drug ran out, and he died a few days later. However, better results were obtained with other patients, and soon there were plans to make penicillin available to British troops on the battlefield. Pharmaceutical and chemical companies played a particularly important role in solving the problems associated with scaling submerged fermentation from a pilot plant to industrial manufacturing scale. As production increased in size, scientists at Merck, Pfizer, Squibb, and other companies faced new engineering challenges. Pfizer's John L. Smith was able to portray the complexity and uncertainty these companies faced during the scale-up process. Mold is as temperamental as an opera singer. Yields are low, isolation is difficult, extraction is murder, purification invites disaster, and testing is unsatisfactory. Because penicillin requires air to grow, aerating the fermentation mixture in such deep tanks posed a problem. When corn syrup was first used as a culture medium, bubbling sterile air through the mixture caused foaming. Squibb solved this problem by introducing an anti-foaming agent, glyceryl monoricinoate. On March 1, 1944, Pfizer opened the first commercial plant for the large-scale production of penicillin by submerged culture in Brooklyn, New York. Meanwhile, clinical studies, both military and civilian, confirmed the therapeutic promise of penicillin. The drug was shown to be effective in treating a wide variety of infections, including infections caused by streptococcus, staphylococcus, and gonococcus. The United States Army valued the use of penicillin in the treatment of surgical and wound infections. Clinical studies also demonstrated its effectiveness against syphilis, and by 1944 it was the primary treatment for this disease among the armed forces of Great Britain and the United States. Daniel 12:4. But you, Daniel, shut up the words, and seal the book until the time of the end. Many shall run to and fro, and knowledge shall increase. Glories and praises to the God of Israel. Maranatha. Acknowledgements. Adapted for the Internet from the discovery and development of penicillin 1928 to 1945. Prepared by the American Chemical Society and the Royal Society of Chemistry, United Kingdom, in 1999.